A very good afternoon to everyone. Welcome to today's class. And we are going to talk about blood glucose level regulation in today's class. So that is the topic of the class, blood glucose level regulation. But before beginning, let us have a small introduction about Unacademy. And as we say in the starting that let's crack NEET PG together. So those students who are watching the class live now, please reply in the comment box if my voice is audible and it's clear. So the topic of today is blood glucose level regulation. But blood glucose level regulation is the topic of today. Please reply in the comment box if my voice is audible. Then Unacademy is one of the India's largest online platform for the preparation of NEET PG examination. India's best faculty members, they teach at Unacademy, so you can get this benefit of being mentored by the best faculty member all over India. Then there are special classes, and what is so special about these special classes is you can ask your doubts and queries directly with the faculty member. They'll be very much willing to solve your doubts and queries. Then there are test series and there are free mock tests, weekly tests, and with the help of these tests you will be able to evaluate how much you have learned so far. And I always say, for qualifying any kind of competitive examination, it is very important to go for the various PYQs and MCQs for understanding the pattern of examination. Then it is very easy to get the app. Just go on Play Store and write Unacademy Learning app. You will see the logo of Unacademy Learning app, and you can just download the app, log in, make your account, and you're good to go. So the way of preparation through Unacademy is very flexible. There is flexibility in the schedule. Then the language can be of your choice and unlimited practice. Once a quiz is taken, it can be retaken as many number of times as much you want to. Then if a video has been delivered live, it is always recorded and you can anytime go back and watch the video. Then you won't feel anything different than a normal classroom because you can chat with your educator. Then there are interactive polls. Then you can learn anytime, anywhere. And in interactive polls, what will happen that you will be given some 4 or 5 options. So you will have to select one option, and you will also come to know why other options are not suitable for the answer, why the other options are wrong, because the faculty members, they'll be explaining you each and every option in detail. So you can chat with your educator on this platform. Then there are interactive polls, and the greatest benefit is you can learn anytime, anywhere. If you're not in town or if you are somewhere busy, then you can anytime go back and watch the video. So it is like learn anytime, anywhere kind of concept. Then there is this plus subscription, and in this plus subscription you will get all the 19 subjects, each and every topic in a very sequentially recorded manner. So go for this plus subscription and prepare for your examination, because there the faculty members, they keep on taking the short-term courses, then crash courses for all the upcoming examinations, seeing the pattern of examination, the courses are being designed. So plus subscription can be very much beneficial for your qualifying the examination. And for this plus subscription you need to pay a certain amount for the plus subscription, and that is like quite pocket friendly. If you are going for a 12 monthly or 2 yearly program, then it is very reasonable and pocket friendly as well, and you will also be able to cover all the topics which are going to be asked in the examination. And For getting a 10% discount, just use my referral code FB01. By using my referral code FB01, you will be getting a 10% discount in the Plus subscription. So do not forget to use my referral code, that is FB01. So starting with the topic of today, that is blood glucose level regulation. Blood glucose level regulation. So I hope my voice is quite clear and audible. Let us talk about it. So first of all, what is the normal blood glucose level? So normal blood glucose level is 70 to 110 milligram per dl, and this 70 to 110 milligram per dl is for the fasting state. Is for the fasting state, that is 10 to 12 hours after meals or overnight. 10 to 12 hours after meals or overnight. Then what is the post glucose load? Post glucose load. So the range for post glucose load is 110 to 140 milligram per dl. So this is for 2 hours after 75 gram of glucose is administered. So after 2 hours. After 2 hours of the administration of glucose. So first is normal glucose level, second is post glucose load, and the third is random. The range of random is 70 to 140 milligram per dl, and this is the normal plasma glucose level. This is normal plasma glucose level, and it is done by glucose oxidase peroxidase method. By glucose oxidase peroxidase method. Random. Then there are different sources of blood glucose, and there are different places where they are utilized. Different places of their utilization and different sources. So talking about the different sources of blood glucose and their utilization. So first of all, talking about the sources. Talking about the sources. So the important dietary source of glucose is carb. Important dietary source of glucose is carb or carbohydrates after digestion and absorption. So after digestion and absorption, carbohydrate, they are a very important source of dietary glucose. Second, in the body there is generation of glucose through gluconeogenesis. Generation of glucose in body through gluconeogenesis. Right? So what is this gluconeogenesis? That means synthesis of glucose from non-carbon sources. So the non-carbon sources which contribute in the blood glucose level, so they are lactate, glycerol, then propionyl CoA, then glucogenic amino acids. Glucogenic amino acids and except leucine. So almost all of the amino acids will be included except leucine. So this is the source of glucose in the blood, gluconeogenesis from the non-carb sources. Here one should mention non-carbohydrate source. First one was dietary from carbohydrate, second is non-carbohydrate source through gluconeogenesis, and another source of blood glucose level is glycogenolysis. Glycogenolysis. So that means breakdown of glycogen into glucose. Breakdown of glycogen into glucose. Glycogen into glucose. This is the third source, and it is a very important source of glucose in between the meals. It's an important source of glucose in between meals. In between meals, glycogen will contribute to glucose, and glucose is required for the activity of brain and RBCs. So blood glucose level in between the meals, it is maintained by glycogenolysis. Right? Now moving forward and talking about the utilization of blood glucose. Right? So utilization of blood glucose. Utilization of blood glucose. Where are they utilized actually? So the first place is glycolysis, TCA cycle. So by glycolysis and TCA cycle, the blood glucose level, the blood glucose is converted into carbon dioxide and ATP. So the first place is glycolysis. The second place is HMP shunt. Now the third place where it is used is in well-fed state, when the conversion of glycogen occurs from glucose. So glucose is used in formation of glycogen in well-fed state. And the process of formation of glycogen from glucose is called as glycogenesis. So glycogenesis. Then the fourth place where it is used is in the lipid synthesis. So lipogenesis. Lipogenesis or lipogenesis, that is the formation of lipids. Then it is used for the glycolipids formation, synthesis of glycolipids, then amino sugars, etc. So these are the places where the blood glucose level is used up. Now whenever the glucose level, it crosses the renal threshold, and that is 180 milligram per dl, it appears in urine if it crosses the renal threshold. So renal threshold. it crosses the renal threshold, that is 180 milligram per deciliter, what will happen? It will appear in urine. Appear in urine, right? So when glucose it appears in urine, the condition is called as glycosuria. When urine when in urine glucose appears, it is called as glycosuria, and this occurs because of the high carbohydrate diet and other renal causes. So this occurs because of high carb diet and various other renal causes and etc. So 180 milligram per deciliter is the renal threshold for the blood glucose. Now regulation part, for which we are starting this. So regulation of blood glucose. Regulation of blood glucose. Regulation of blood glucose. So the important hormones which are required for the regulation of glucose in blood is insulin and glucagon. So the two most important hormones are insulin and glucagon. These are the most important ones. We'll be talking about some other also. So whenever blood glucose level is low, it is called as low blood glucose level, is called as hypoglycemia. Whenever it will be high blood glucose level, it is called as hyperglycemia. So the hormone, or the only hormone which reduces the blood glucose level, is insulin. So let us write only hormone which reduces blood glucose level is insulin. The only hormone which is responsible for reducing the blood glucose level is insulin. Now there are hormones which are participating in increasing the blood glucose level. So let us see the names of the hormones which are responsible for increasing the blood glucose levels. So let us write hormones which increases blood glucose level. So the name of hormones which increases the blood glucose level is glucagon, then it is epinephrine, then it is thyroxine, then it is growth hormone, then cortisol, then adenocorticotropic hormone, ACTH. These are the hormones which actually increases the blood glucose level. So this glucagon is mostly affecting the liver, epinephrine, liver and muscles. So rest all the hormones, they are responsible for increasing the blood glucose level. So they are in total 6 hormones which contribute in increasing the blood glucose level. They increase the blood glucose level and they are responsible for hyperglycemia. Responsible for hyperglycemia. right? Now talking specifically about insulin. Insulin. Talking about insulin. So first thing, it increases the uptake of glucose by cells. Increases uptake of glucose by cells by insulin dependent transporters. By insulin dependent transporters. So the example of insulin dependent transporters, one of the example is GLUT4. So it increases the uptake of glucose by cells. Second, it increases glycolysis. Increases glycolysis and HMP shunt. HMP shunt increases glycolysis and HMP shunt. Then it decreases gluconeogenesis. Decreases gluconeogenesis. Then it inhibits glycogenolysis. Inhibits glycogenolysis. So its action is it increases the uptake of glucose, increases glycolysis and HMP shunt. It decreases gluconeogenesis and it inhibits glycogenolysis. Right? So this was about insulin. Talking about the hormones which actually increases the blood glucose level or which are responsible for hyperglycemia. These hormones. Okay, let us go to that page. Yes. So these hormones, they increase the blood glucose level by stimulating the glycogenolysis. So how do they do? They stimulates glycogenolysis and it stimulates glucogenesis. Gluconeogenesis. In a better way, we should write gluconeogenesis. Gluconeogenesis. So stimulates glycogenolysis, that is breakdown of glycogen into glucose, and gluconeogenesis, that is formation of glucose from the non-carb sources. So stimulates glycogenolysis and stimulates gluconeogenesis. Right? Now talking about the mechanism of regulation, that what is the mechanism of regulation? So mechanism of regulation. Mechanism of regulation. So regulation of glycogen metabolism by insulin and glucagon, that we are going to see. So let us write over here. Regulation of glycogen metabolism by insulin and glucagon. This we are going to see. So for glycogenesis, glycogen synthase is responsible, or the key enzyme is glycogen synthase. And for glycogenolysis, glycogenolysis, the key enzyme is glycogen phosphorylase. Glycogen phosphorylase. So for glycogenesis, it's glycogen synthase. For glycogenolysis, it's glycogen phosphorylase. So insulin controls these two enzymes through phosphorylation and dephosphorylation. So insulin, it controls both of the enzymes through phosphorylation and dephosphorylation. So glycogen synthase, it is active in dephosphorylated state, and glycogen phosphorylase is active in phosphorylated state. So glycogen synthase in dephosphorylated state and glycogen phosphorylase in phosphorylated state. Okay, so glycogen synthase should be active in dephosphorylated state and glycogen phosphorylase is active in phosphorylated state. Fine. So insulin, it controls both of the hormones. So what insulin will do? Insulin will decrease the blood glucose level and glucagon will increase the blood glucose level. So what will happen? This insulin, it is decreasing the blood glucose level. It means it is responsible for dephosphorylation of glycogen synthase, so that it becomes active and it performs glycogenesis, and glycogenolysis, it will stop glycogenolysis because it does not want the breakdown of glycogen into glucose. So it will keep glycogen phosphorylase in dephosphorylated state again, so that there should be no breakdown of glycogen, right? So moving ahead and talking about the pathway of regulation. Talking about the pathway of regulation. Pathway of regulation. So what happens over here is this glucagon. Glucagon, I told you previously also, it's a hormone which is responsible for increasing the blood glucose level, and mostly its action is on liver, and epinephrine and glucagon, they together act on liver and muscles. So glucagon and epinephrine attaches to the receptor. How they are regulating the blood glucose level? They attaches to receptor on the cell, and they act via cyclic AMP. They act via cyclic AMP. Remember, their mode of action or the secondary messenger is cyclic AMP, and they cause the formation of adenylate cyclase. Causes formation of adenylate cyclase. Adenylate cyclase. And this converts ATP into cyclic AMP. This converts ATP into cyclic AMP, and cyclic AMP activates protein kinases. Activates protein kinases. Activates the protein kinases, and protein kinases, they activate phosphorylase kinase. Protein kinase activates phosphorylase kinase. Phosphorylase kinase, and this now activates glycogen phosphorylase via phosphorylation. Activates glycogen phosphorylase. Phosphorylase via phosphorylation. So this glycogen phosphorylase is the key enzyme in glycogenolysis. So that means there is stimulation of glycogenolysis. Glycogenolysis will occur. Right? So let us just have a very quick revision of the pathway of regulation through glucagon and epinephrine. Glucagon mostly act on liver, epinephrine acts on liver and muscle. They attach to the receptors on the cells and they act via the secondary messenger, that is the cyclic AMP, which will lead into formation of adenylate cyclase, and the role of adenylate cyclase is to convert ATP into cyclic AMP. And when ATP gets converted into cyclic AMP, what will happen? They will activate the protein kinase, and the protein kinases, they activate the phosphorylase kinase, and phosphorylase kinase, they perform the phosphorylation of the key enzyme of glycogenolysis, that is glycogen phosphorylase. And glycogen phosphorylase, when it is phosphorylated, it performs glycogenolysis. Thus, there is increase in the blood glucose level. Right? So, this was how the glucagon and epinephrine, they control the blood glucose level. Now, talking about calcium. So, calcium also activates glycogen phosphorylation, and it increases the blood glucose level, by activating glycogen phosphorylase. So it also activates the glycogen phosphorylase, right? So protein kinase, it phosphorylates glycogen synthase and inactivates it, thus no glycogen formation from glucose. So here this protein kinase, it has one more function, and its one more function is to perform the phosphorylation of glycogen synthase, and like this, by its phosphorylation, it will be inactivated and there will be no formation of glycogen. No glycogen formation, because glycogen synthase is now inactivated and it is the key enzyme of the glycogen synthesis or glycogenesis, right? So we have seen how insulin acts, we have seen how glucagon and epinephrine it works. Now let us see that mechanism of action of insulin, which causes the utilization of cyclic AMP. So mechanism of action of insulin, which causes utilization of cyclic AMP. Mechanism of action of insulin, which causes the utilization of cyclic AMP. So cyclic AMP, it gets converted into 5-AMP. It gets converted into 5-AMP by the help of enzyme phosphodiesterase. Cyclic AMP, it gets converted into 5-AMP by the help of phosphodiesterase. Now, this cyclic AMP, it inactivates the protein kinase. Thus, glycogen synthase is in dephosphorylated state and active glucose to glycogen and activates the synthesis of glycogen from glucose. So, cyclic AMP inactivates the it will inactivate the protein kinase, and thus glycogen synthase is in the dephosphorylated state, and thus glycogen synthesis will be suppressed, and it will also inhibit the glycogenolysis. Also inhibit glycogenolysis. Also inhibit glycogenolysis, right? And insulin, it inhibits glucose formation, and glucagon will favor glucose formation, that we have been seeing since starting. Now talking about regulation of gluconeogenesis. Regulation of gluconeogenesis. Regulation of gluconeogenesis. So formation of glucose from non-carbohydrate precursor is called as gluconeogenesis. Formation of glucose from non-carb precursor called as gluconeogenesis. Right? So there are 4 key enzymes of gluconeogenesis. 4 key enzymes of this. What are these 4 enzymes? Let us revise the names of these enzymes. That is pyruvate carboxylase, pyruvate carboxylase. Then it is phosphoenol pyruvate carboxykinase, phosphoenol pyruvate carboxykinase. Then it is fructose 1,6-bisphosphate, fructose 1,6-bisphosphate. Then it is glucose 6-phosphate, glucose 6-phosphatase, phosphatase. Here also fructose 1,6-phosphatase. So the 4 key enzymes of gluconeogenesis is pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6-bisphosphatase, and glucose 6-phosphatase. Right? So this we are going to see. It occurs through 2 ways, that is induction and repression. So this regulation goes by 2 things, that is induction and repression. Induction and repression. So insulin, it is the inhibitor or repressor, and glucagon is the inducer. So in what happens is, talking about insulin, it represses. Insulin represses pyruvate carboxylase. Pyruvate carboxylase. Insulin, it represses pyruvate carboxylase, and glucagon, it induces pyruvate carboxylase. This pyruvate carboxylase. So glucagon is inducer of pyruvate carboxylase. Now talking about some other things, that is acetyl-CoA. Acetyl-CoA, it induces pyruvate carboxylase. Citrate, it induces fructose 1,6-phosphatase. Fructose 1,6-bisphosphatase. So acetyl-CoA and citrate. Then there are 3 other important enzymes. 3 other important enzymes. So first one is glucokinase and hexokinase. Glucokinase and hexokinase. The second one is phosphofructokinase. phosphofructokinase. Then the third is pyruvate kinase. Pyruvate kinase. So insulin, talking about insulin, insulin is inducing the pyruvate kinase. It induces the pyruvate kinase and thus it favors the glucose utilization and favors glucose utilization. Then glucagon, what does it do? It inhibits the pyruvate kinase. Inhibits pyruvate kinase and it does not favor the utilization of glucose. So glucagon is negative inducer of pyruvate kinase, right? Now talking about status of glucose in different states. So talking about status of glucose in different states, like in well-fed or starvation and like that. So first one is in well-fed state or after meals or well-fed state, after meals or well-fed state. So what happens after meals? The level of glucose is high. The level of glucose is high. So glycogen synthesis occurs. Glycogen synthesis occurs. Then the source of energy in brain and in RBCs is glucose. So glucose is source of energy. Glucose is source of energy in brain and RBC. So the three things: glucose level is high, glycogen synthesis occurs, and glucose is the source of energy in brain and RBC in the well-fed state. Now the second thing, the second state is post-absorptive state. Post-absorptive state, and it is also called as in between the meals. In between meals. So the glucose level is mainly maintained by liver by gluconeogenesis. So glucose level is maintained by glycolysis, by liver. Okay, so in between the meals, the level is maintained by liver by glycogenolysis. Glycogenolysis, that is the breakdown of glycogen into glucose. That is the breakdown of glycogen into glucose. Now talking about the third state, that is the after meals or after 16 to 18 hours after meals. After 16 to 18 hours after meals or in starvation, when the main source of glucose is gluconeogenesis, or main source of glucose is gluconeogenesis. So this gluconeogenesis means formation of glucose from non-carbohydrate source. So it occurs in liver. And What happens in this? In this, there will be utilization. There will be utilization of amino acids from muscles and brain. So utilization of amino acids from muscles and brain will start using ketone bodies as energy source. Brain will start using ketone bodies as energy source. To some of the extent. So this is the state of ketosis. The third is, that is 16 to 18 hours after meals, and that is the main source of glucose. Right? Now the fourth state is after 3 days of starvation. After 3 days of starvation. After 3 days of starvation, what will happen? Amino acids and proteins of skeletal muscles is utilized for glucose production. And brain solely depends on the ketone bodies as the source of energy. And brain depends on ketone bodies as source of energy. So after 3 days, there will be utilization of the amino acids and proteins of skeletal muscles, and this will lead to the production of glucose. This will lead to production of glucose, and the brain will get its energy from the ketone bodies. Right? Now moving a little forward in this, that is talking about estimation of plasma glucose. Estimation of plasma glucose. Estimation of plasma glucose. So this is mainly done by glucose oxidase peroxidase method. Mainly done by glucose oxidase peroxidase method, which is also called as GOD POD method. So GOD POD method. So nowadays this is replaced by hexokinase method. Nowadays replaced by hexokinase method. So I can see some of the students live. So please, they can reply in the chat box. Are they able to understand? So nowadays it is replaced by hexokinase method. So let us talk about it, that whenever there will be increase in blood glucose level, that means it is hyperglycemic condition, hyperglycemia, and this hyperglycemia is because of diabetes mellitus, Dipansh Nair is saying yes. Ayush Shukla is saying yes. Okay, thank you for your comments. Then the second thing which will increase in the blood glucose level is overactive thyroid. Overactive thyroid or hyperthyroidism. Hyperthyroidism. Then the third is hyperpituitarism. Hyperpituitarism. And the fourth is hyperadrenalism. Hyperadrenalism. Dipansh Nair is saying that, ma'am, please start with molecular biology concepts. So, molecular biology, I have already completed last month. Have you gone through my videos on replication, transcription, translation, then repair mechanism, protein folding? These all videos I have already done, Dipansh, with molecular biology, these topics of molecular biology. So any specific topic which you want to read, you just write in the comment box. Have you gone through my videos? So these videos on the molecular biology topic, it is already there on this channel. Please reply. Then what are the causes of hypoglycemia? No, ma'am. Okay, so just try to go through those videos. I have completed the series on replication, transcription, translation, post-transcriptional modification. I've done splicing. Then lots of videos are there. Just try and see those videos. And if you come across any kind of doubt, you can just write in the comment box whenever I am going live, or in the comment box you can write so that I can understand that what else you want to read. And other than molecular biology and the videos which I have already done, just try to see what you want to study, so that in next class I'll try to cover that. So finally, what are the causes of hypoglycemia? Hypoglycemia, the causes of hypoglycemia. So incorrect dose of insulin therapy. Incorrect dose of insulin therapy. Then less intake of food, decreased intake of food. Then hypoactivity of pituitary, adrenal and thyroid. Hypoactivity of pituitary, adrenal and thyroid. So like this, we have covered the topic which we wanted to cover today, that is the blood glucose level regulation. So for your convenience, I'll just go through the whole topic quickly, or we'll do a very quick revision. So the topic that we were reading in today's class was blood glucose level regulation. So the normal blood glucose level is 70 to 110 milligram per dl. So this 70 to 110 milligram per dl, it is for 10 to 12 hours of fasting or overnight. Then the post glucose load, that is after the 2 hours of administration of 75 gram of glucose, that is 110 to 140 milligram per dl. Then random level of blood glucose is 70 to 140 milligram per dl. So this normal plasma glucose level, we have just seen that it is usually measured by glucose oxidase peroxidase method, GOD-POD method. So coming to the sources of blood glucose, so dietary source, that is carb, is the most important source of glucose. Then the glucose is generated by gluconeogenesis through the non-carbon sources from lactate, glycerol, propionyl CoA, glucogenic amino acids. Then glycogenolysis, that means breakdown of glycogen into glucose. So it is the source of energy in between the meals. So secondly, the source of glucose is glycogenolysis. Then utilization of blood glucose, where is it utilized? So it is utilized in glycolysis and tricarboxylic acid cycle, and it gives rise to carbon dioxide and ATP. It is utilized in HMP shunt. Then it is utilized in glycogenesis, that is for the formation of glycogen in the well-fed state. Then it can be used for lipogenesis, like for the formation of glycolipids, for the formation of amino sugars. Then the renal threshold for the blood glucose level is 180 milligram per dl, and if it crosses this threshold, it will start appearing in urine, and the condition is called as glycosuria. And the glycosuria occurs because of the high carb diet and various other causes such as some of the renal causes. Then we have seen the regulation of blood, how insulin and glucagon, they play the key role. So the low blood glucose level is called as hypoglycemia, and high blood glucose level is called as hyperglycemia. So the only hormone which is responsible to reduce the blood glucose level is insulin, the only hormone. Then the hormone which is responsible for increasing the blood glucose level, so we have seen three. So first one is glucagon, second is epinephrine, third is thyroxine, third is growth hormone, fifth is cortisol, sixth is actinocorticoid tropic hormone. And what they do, they stimulate the glycogenolysis, that is breakdown of glycogen into glucose, and gluconeogenesis, that is the formation of glucose from the non-carb sources, which we have just seen now, the four of the non-carb sources. Then how the insulin it acts? Insulin, what does it do? It increases the uptake of glucose by the cells, and it increases the increases in the expression of the insulin dependent transporters, that is the one of the example is GLUT4. Then insulin, it increases the glycolysis and HMP shunt. It decreases the gluconeogenesis from the non-carb sources. It inhibits the glycogenolysis, that is breakdown of glycogen into glucose. Then what is the mechanism of regulation? So mechanism of regulation of insulin and glucagon. So for glycogenesis, the key enzyme is glycogen synthase. For glycogenolysis, the key enzyme is glycogen phosphorylase. So glycogen synthase is active in dephosphorylated state, and glycogen phosphorylase is active in the phosphorylated state. So what is the pathway of regulation? The glucagon is mainly affecting the liver, and epinephrine is mainly affecting the liver and muscle. So what will happen? This glucagon and epinephrine, it will attach to the receptors on all the cells, and it will increase the cyclic AMP concentration, that is the secondary messenger, and it acts via cyclic AMP, and it causes the formation of adenylate cyclase. So there will be formation of adenylate cyclase, and adenylate cyclase, what it will do, it will convert ATP into cyclic AMP, and cyclic AMP will activate protein kinase. Protein kinase will activate the glycogenolysis, and it will inhibit the gluconeogenesis. So this is how insulin will decrease the level of glucose. Then mechanism of action of insulin is done by the cyclic AMP. It will lead into formation of 5-AMP through the enzyme phosphodiesterase. So it will inhibit the glycogenolysis, and it decreases the glucose level. Then regulation of gluconeogenesis. So there are four key enzymes, that is pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6-bisphosphatase, and glucose 6-phosphatase. So these are the key regulatory enzymes of this. So insulin, it represses pyruvate carboxylase, and glucagon, it induces the pyruvate carboxylase. Acetyl-CoA, it induces pyruvate carboxylase, and citrate, it induces the fructose 1,6-bisphosphatase enzyme. Then the three other important enzymes are glucokinase, hexokinase, phosphofructokinase, and pyruvate kinase. Insulin, it is the positive regulator of pyruvate kinase. Glucagon, it is the negative regulator of pyruvate kinase. Now, status of glucose in different state. After meal, the level of glucose is high, glycogen synthesis occurs, glucose is the source of energy in brain and RBC during the well-fed state. In between the meals, glycogen is the source of energy, and In case of 16 to 18 hours of fasting, gluconeogenesis will occur in liver, and it will lead to utilization of amino acid from the muscles, and brain will start using the ketone bodies to some extent. In case of 3 days of starvation, amino acids and proteins of the skeletal muscle is utilized for glucose production, and brain, it depends on the ketone bodies mainly for the energy 3 days after starvation. Then what is how the estimation of plasma glucose occurs? So it is by the GOD-POD method, that is glucose oxidase peroxidase method, but these days it is done by hexokinase method mostly. Then whenever there will increase in the blood glucose level, it is called as hyperglycemia. It is because of diabetes mellitus, it can be because of hyperthyroidism, it can be because of hyperpituitarism, it can be because of hyperadrenalism. Then what is the causes of hypoglycemia? So incorrect dose of insulin during the insulin therapy, when there is decreased intake of food, and when there is hypoactivity of pituitary, adrenal and thyroid. So like this, we have completed all the topics in the blood glucose level regulation. So like this comes to the end of the lecture, and please subscribe our channel, that is Let's Crack NEET PG. Please hit the bell icon for getting all the notifications of the upcoming videos. Also use my referral code, that is FB01, for getting 10% discount on the plus subscription. So please don't forget to use the code FB01, and also please give a thumbs up if you have understood the concept and if you have liked the content which I wanted to explain to you. So this is it for today's class. We'll be meeting some other day, maybe tomorrow, for the next topic related to the biochemistry. So this is it for today's class. Have a very good day. Keep on studying.